It's awesome to see so many friends, family, and colleagues all here together in person, and we've got quite a few people on the Zoom too. So thank you all so much for being here. I am very excited to share with you my PhD thesis work on immune-acting fibroblasts, or IAFs, a cell type which we are learning plays a very key role in skin inflammation and host defense. Now, before I get started, I really need to give a huge thank you to my committee members, not just Rich, but also Mick, Chris, Manuela, Victor. I'm really fortunate to have such an all-star cast of scientists guiding me, so thank you so much for your guidance. So what is a PhD thesis? Well, according to Google, a thesis is a statement or theory that is put forward as a premise to be maintained or proved, or a long essay or dissertation involving personal research written by a candidate for a college degree. I think both of these definitions apply well to a PhD thesis. Over the course of the next 45 minutes, I'm going to share with you my personal research on the immune system. And then at the end, I'm going to conclude by putting forth my theory about how that system works. So we're going to start today by learning about the skin and how it defends us against a pathogen called Staphylococcus aureus, or Staph aureus. If you've heard of staph infections before, this is what we'll be talking about. We'll then spend the bulk of our time today talking about our lab's recently published discovery of immune-acting fibroblasts, or IAFs, that defend us against Staph aureus infections, but also contribute to psoriasis pathogenesis. In the second portion of the talk, I'll share with you some exciting unpublished work where we're using these really cutting-edge transcriptomic technologies to understand a class of diseases called neutrophilic dermatoses. And using these approaches, we've identified a role for immune-acting fibroblasts in a condition called Sweet syndrome. And finally, we'll wrap up with some conclusions, talk about where future research efforts should be invested, move on to the Q&A. I'll tell some fun stories about how I got to where I'm at and thank the people who helped me along the way. And then the party if I pass, right? So we have a party. Okay. So Staphylococcus aureus, or Staph aureus, is the bacterial pathogen that is responsible for most skin and soft tissue infections in humans. Here's a 20,000 times magnified view of Staph aureus. Each purple sphere here is a single bacterium. It's not actually purple, it's just pseudo-colored. And here is an example of a skin infection that might be caused by Staph aureus. Now, by a show of hands here, who has had a staph infection before? Anyone? Okay, that's a lot of us, right? It's pretty common. I've had a few myself. And in most cases, you take antibiotics and the infection clears right up, no problem. However, in some cases, most of our antibiotics won't work. And this is because some staph strains have evolved resistance to most of the antibiotics in our arsenal. This makes treatment extremely complicated, or even in some cases impossible, and leads to a lot of morbidity and mortality. In fact, in the US alone, MRSA causes around 20,000 deaths annually. MRSA is the antibiotic resistant strains here. And this is more than any other bacteria and on par with the flu virus. However, we may be able to address this global health concern if we could better understand how our skin naturally protects us against Staph aureus. Because with this knowledge, we could develop host-directed therapeutics, drugs that boost our immunity to combat the infection. So what do we know so far about how the skin defends against Staph aureus? Well, first, let's just talk about the skin. So we can divide the skin up into two major parts, the epidermis at the surface of the skin and the dermis underneath. And the epidermis is composed of layers and layers of cells called keratinocytes. And when your skin encounters Staph aureus, your keratinocytes are among the first cells to recognize and detect the Staph aureus. And in response, they can produce molecules called antimicrobials. These are natural antibiotics that your body already makes on its own to kill the staph. Additionally, keratinocytes can ring the alarm bells and tell other cells to come help with this infection. And they do this, for example, by secreting molecules called chemokines. These are protein messengers that attract other cells into the skin. In particular, these cells make neutrophil chemokines that attract a cell type called a neutrophil into the skin from the blood and bone marrow. They don't normally live in the skin, but they come in to help. And these cells are very potent killers of Staph aureus. And we know how important they are because when patients are neutropenic, when they lack neutrophils, they're at very high risk for severe disease. These cells are like the Swiss Army knife of the immune system. They have many different weapons to help kill the staph. They can release extracellular traps to immobilize the Staph aureus, like Spider-Man. And they can also release reactive oxygen species and antimicrobials to actually kill the pathogen. And they can also just eat or phagocytose it. So very potent killers of Staph aureus, the neutrophils are. Another cell type that is very important for defense against staph is the lymphocyte, in particular the subsets that produce the cytokine IL-17. And a cytokine is just another protein messenger of the immune system. It tells other cells what to do. And IL-17 is a very potent amplifier of defense against Staph aureus. It acts on those keratinocytes in the epidermis and boosts up their production of antimicrobials and boosts up their production of chemokines, leading to a ton of neutrophils coming into the skin. So it's really amplifying the host defense response. Now, if you talk to an immunologist about how the skin defends against Staph aureus, these are the three major cell types you're going to hear about: keratinocytes, neutrophils, and lymphocytes. However, there are a lot of other cells in the skin that may contribute to defense, and an opportunity to advance treatment may come from more research on these other cell types. In 2015, a postdoc from our lab, Ling Zhang, made a novel discovery in regards to host defense against Staph aureus when she showed that during infection, subsets of cells in the dermis called fibroblasts would differentiate into fat cells or adipocytes. This is they're transforming into adipocytes. And importantly, she observed a huge increase in adipose tissue in the skin in just a couple of days. And this was important because she observed that cathelicidin was secreted by these cells while they were differentiating. And this antimicrobial peptide cathelicidin is very important for killing Staph aureus. So really, she identified a new player in host defense, the immature adipocyte. So when I joined the lab in 2020, I contributed to ongoing projects, including this one led by postdocs Mark Liggins and Alan O'Neill, showing that reactive adipogenesis also occurs in the very common human skin disease, acne. Importantly, we showed that the very effective treatment for acne, Accutane, actually works by boosting the antimicrobial production during reactive adipogenesis. Now, when I joined the lab, I became very interested in the fibroblast itself, while everyone else was focused on the fat cells. And I was interested in these cells because they're the most abundant cell type in the dermis, and they have historically been overlooked in terms of immunity. What they are appreciated for is secreting something called extracellular matrix. And this is like the glue that holds our tissue together, gives it structure and architecture. Fibroblasts are also appreciated for their role in wound healing. So when you have an injury or a cut in your skin, some of your fibroblasts will turn into a special type of cell called a myofibroblast that makes a lot of this matrix to promote scar formation. We know in the skin that there are two spatially defined lineages of fibroblasts. We have the upper lineage of fibroblasts close to the epidermis called the papillary fibroblasts, and then the reticular fibroblasts that are deeper in the skin. And as I alluded to, some reticular fibroblasts are pre-adipocytes that can turn into fat cells. So one of the first things I did when I joined the lab was I started synthesizing all of the literature related to fibroblast immune function at barrier tissue sites. So not just only the skin, but also the gut and the lung, places where infections commonly start. And I wrote this review with Rich, and we were really surprised to find that there were actually decades of studies, mostly in vitro in a dish, showing that fibroblasts could respond and recognize microbes and respond to pro-inflammatory cytokines, and in turn elicit functions that may be relevant in immunity. So over the course of writing this review, we really started changing how we were thinking about fibroblasts. Instead of these cells being merely structural cells that make matrix and turn into fat, we now recognize that these cells are incredibly diverse. There's many different types of fibroblasts. And we recognize this in light of the in vitro studies that I've mentioned, as well as some more recent single-cell multiomic studies. And importantly, these studies have highlighted that there's functional heterogeneity of these cells, and some of them may be important in immunity. However, we don't know if dermal fibroblasts as a whole or any subset play a significant role in skin inflammation and host defense. And this is what I set out to address with my thesis. So in this first part of the talk, I'm going to share with you our recent discovery of immune-acting fibroblasts, or IAFs, that defend us against Staph aureus infections, but also contribute to psoriasis pathogenesis. And this work was recently published in the Journal of Experimental Medicine, so please feel free to check out the article for the full story. And I'm also very excited to announce we just learned last week that our article will be featured on the cover of the journal in the next issue. So in this report, we asked the question, how does the skin defend against Staph aureus? And what we could have done was we could have just focused in on the fibroblast, but this is a biased approach, and it's equivalent to looking for your keys under the lamppost because that's where the light is, not where you lost your keys. So we prefer to start with unbiased methods, and instead we looked at all of the cells. And this is more equivalent to the Eye of Sauron in Lord of the Rings that sees all. Some people get it. So we did this by infecting mice intradermally with a MRSA strain of Staph aureus right into the back skin. And then we performed a technology called single-cell RNA sequencing. And this approach allows us to look at all of the cell types in the skin and all of the genes that they are expressing. We can then use these data to perform a network analysis, this machine learning analysis, that lets us see which cells are talking to each other during infection. And what this analysis revealed was a dominant fibroblast to myeloid communication pathway. And note that neutrophils, those really potent antimicrobial cells, are a type of myeloid cell. So this is really surprising to us because based on current models of pathogenesis, we expected the keratinocyte to myeloid communication would have been the strongest. But no, it's dominant fibroblast. So perhaps the fibroblasts are recruiting neutrophils for host defense? Now, to understand how the fibroblasts might be communicating with myeloid cells, we looked at the differentially expressed genes in the fibroblasts during infection. And we found that these cells upregulate many host defense genes. Importantly, we see neutrophil chemokines, these CXCL molecules, pro-inflammatory cytokines, antimicrobials, and other genes. And overall, these genes were most consistent with the IL-17 signaling pathway. And remember that IL-17 is that key amplifier of defense against Staph aureus. Additionally, relative to other cell types, fibroblasts highly express these neutrophil chemokines. They also just outnumber the other cell types. So these cells are a major source of molecules that attract neutrophils. So we hypothesized based on these data that IL-17 may also activate fibroblasts, not just keratinocytes, to recruit neutrophils for host defense against Staph. Now, to understand whether a specific fibroblast subset is talking to neutrophils, we looked at the subsets of fibroblasts in our data, and we resolved 11 transcriptionally distinct clusters. And we found that clusters 1 through 7 on the right of the dotted line highly express neutrophil chemokine, whereas those on the left of the dotted line, 8 through 11, largely did not. Additionally, we observed that some chemokines like CXCL12 were expressed very broadly across these clusters, whereas others like CXCL5 were expressed by a much smaller subset of cells. So this suggests that there's even different types of fibroblasts that can talk to neutrophils. Now, to characterize these cells further, we looked at expression of known fibroblast subset markers. And we found that clusters 1 through 7 also highly express reticular and adipocyte lineage markers, but not papillary nor myofibroblast markers. Therefore, we've defined the fibroblast subsets as a whole that talk to neutrophils as CXCL12 positive adipocyte lineage. And we confirm this definition through more unbiased methods like GO-term analysis. We can see clusters 1 through 7 expressing fat cell differentiation genes and neutrophil migration genes, but we do not see this in clusters 8 through 11. Additionally, our network analysis shows that clusters 1 through 7 talk to neutrophils much more so than clusters 8 through 11. Now, we next asked, where do these fibroblast subsets exist in space? And to address this, we performed confocal microscopy. And here we can see fibroblasts labeled in green, neutrophil chemokines labeled in red, and the overlap between these two signals in yellow. So what we can see is that during infection, fibroblasts upregulate neutrophil chemokine. We see a lot of this yellow. But this is specifically found in the fibroblasts in the deeper reticular dermis, not in the fibroblasts that are in the more shallow papillary dermis closer to the epidermis and keratinocytes. So these data validate our single cell findings of specific fibroblast subsets that talk to neutrophils and further demonstrate that the CXCL12 positive subsets that do talk to neutrophils occupy a unique spatial niche in the deep reticular dermis. So based on these observational data, we developed a testable hypothesis. CXCL12 positive adipocyte lineage fibroblasts are activated by IL-17 to recruit neutrophils. We then tested this hypothesis in vitro in a dish, and we did this by culturing 3T3L1 pre-adipocyte fibroblasts. This is an adipocyte lineage cell line. And we stimulated these cells with IL-17, among other stimuli involved in host defense, and then looked at expression of neutrophil chemokine as an output. And what this screen revealed is that IL-17 on its own did not induce much neutrophil chemokine expression, though it did potentiate or increase the effect of other signals. In particular, we observed very potent synergy when IL-17 was combined with the pro-inflammatory cytokine TNF-alpha. So to understand further how these signals impact this subset of fibroblasts, we performed bulk RNA sequencing. And in this more unbiased method, we see that IL-17 still does not cause much of a change at the whole transcriptome level, whereas TNF does cause very broad changes. But again, IL-17 potentiates or increases the effect of TNF-alpha. And notably, the effect of these pro-inflammatory cytokines was distinct from adipogenesis, which we had studied previously. Now, looking at specific genes, we can see that fibroblasts, in response to IL-17 and TNF, upregulate many host defense molecules. You can see chemokines, antimicrobials, pro-inflammatory cytokines, et cetera. The genes I'd like to highlight today are the chemokines, and I'd like to highlight them because every single chemokine detected in these data was upregulated by IL-17 and or TNF. So we investigated these further at a protein level using multiplex immunoassay as well as other approaches. And we found that IL-17 and TNF induced the secretion of these chemokines, all but one measured. So these cells are making a lot of molecules that can attract other cell types, neutrophils, among other cell types as well. Now, before I get into the functional significance of these chemokines, I'd like to pause and talk about the mechanism by which IL-17 and TNF activate these cells. So to begin addressing this, we looked back at our bulk RNA sequencing data and found that TNF induced genes related to NF-kappa B signaling. And to formally test the role of these signals inducing NF-kappa B signaling, we performed a Western blot analysis and looked at phosphorylation of canonical NF-kappa B signaling intermediates after addition of IL-17 and or TNF. And we found that TNF induced phosphorylation of these intermediates rapidly within 10 minutes, though IL-17 had no effect, nor did it actually increase the effect of TNF. We performed another analysis. This is a immunostaining for P65, which will translocate to the nucleus when phosphorylated, another NF-kappa B signaling pathway. And we see here again, IL-17 not causing any nuclear staining, though we do see this very strongly with TNF, and really no difference when IL-17 is added on top of TNF. So this suggests that there's another mechanism by which IL-17 and TNF are synergizing in these cells. So, we returned back to our bulk RNA sequencing data to try to get an understanding of what's going on. And we came across this gene in the NF-kappa B signaling ontology called NFKBIZ. And this is an NF-kappa B transcriptional cofactor. And we were very interested in this gene because IL-17 alone induced its expression, making it a potential candidate for synergy. Now, to test the role of NFKBIZ in synergy, we performed a genetic inhibition experiment wherein we used siRNA to knock down NFKBIZ in these cells and then stimulated them with IL-17 and or TNF. And we found that when we did this, we completely lost the synergy between IL-17 and TNF. The expression of neutrophil chemokines and antimicrobials went way down. So this gene is very critical for the synergy in terms of these outputs. Interestingly, however, when we knocked down NFKBIZ, chemokines for other cell types like lymphocytes and monocytes actually went up. So this suggests that NFKBIZ may be an important molecular switch that the fibroblast has that it can use to control which cell types it's talking to. Now, back to the function of these chemokines, now that we know a bit about the mechanism. So to test what these chemokines are actually doing, we performed a trans-well migration assay. And in this assay, we have a well, and that well has a membrane in it. And the membrane is cell permeable. The cells can pass through. In the top of the well, we put mouse bone marrow cells, which include neutrophils, among other leukocytes. And in the bottom of the well, we put fibroblast conditioned media. This is all the stuff that fibroblasts are making, all the stuff that they're secreting. And we use conditioned media from fibroblasts that have been activated by IL-17 and TNF or unactivated. And then we measure the cells that migrate across the membrane toward the conditioned media. This tells us what fibroblasts are recruiting. And when we do this, we can see that fibroblasts, when they're unstimulated, promote some migration of leukocytes across the transwell. But when we first stimulate them with IL-17 and TNF, they promote significantly more migration of leukocytes. Notably, when we use just control cytokines, no fibroblasts, there's no effect. Next, to understand which cell types are actually preferentially migrating toward the fibroblasts, we use flow cytometry. And we can see that fibroblasts are really potently recruiting neutrophils specifically. There's some significant monocyte and lymphocyte migration, but really it's dominantly promoting neutrophil migration. Now, we next want to understand how are fibroblasts recruiting the neutrophils, which chemokines are required. And to test this, we performed the same transwell migration assay, but this time we used that NFKBIZ knockdown approach in addition to it. And here we can see that when we knock down NFKBIZ in the fibroblast, we see a significant reduction of migration of neutrophils toward the conditioned media. So there's some NFKBIZ dependent chemokines that are driving this migration, but there's a handful of those, right? So like which ones specifically? And we addressed that next by another inhibition experiment, this time using pharmacological inhibitors targeting receptors on the leukocytes themselves. And here I'm showing you that we can inhibit simply CXCR2 and CXCR4 on the leukocytes and completely eliminate migration toward the fibroblast conditioned media. So we can totally explain the migration that the fibroblasts are driving here through the chemokines CXCL1, 2, 3, 5, and 12. So while there are dozens of purported neutrophil chemokines, these are the ones that are required for this process in this context. And importantly, these results demonstrate that CXCL12 is not just a great marker of the fibroblast subset that talks to neutrophils, but it is a functional marker. It is actually doing some work. So to summarize what we've learned so far in vitro in a dish, we've learned that IL-17 and TNF, these pro-inflammatory cytokines, synergistically activate adipocyte lineage fibroblasts, driving their expression of this NF-kappa B cofactor, NFKBIZ, allowing the release of CXCR2 ligands and CXCL12 to promote robust neutrophil recruitment. So now we know fibroblasts can do this, but the next question is it important in vivo in the body? Is it significant? And this is what we address next. So to address this, we first developed a very simple model to study neutrophil recruitment to the skin. And here we're injecting recombinant IL-17 and TNF into the mouse back skin. And then we can look at immunostaining of neutrophils in green here and see that IL-17 and TNF promote synergistic recruitment of neutrophils in vivo. So exactly what you'd expect based on the in vitro findings in the dish. We then developed a genetically modified mouse. We call this a conditional knockout. And this mouse lacks the IL-17 receptor specifically in fibroblasts. So only fibroblasts have a defect in their ability to see IL-17. Every other cell type is totally fine. And this is a super important reagent that we developed because if we can compare this mouse to its littermate control that's totally normal, and we see a difference in that model I just described, then we know that fibroblasts and their recognition of IL-17 is important. So this is exactly what we did. We used our new mouse in our new model, and we find that in normal control mice, IL-17 and TNF induce the expression of neutrophil chemokines, but this is suppressed in our mice that lack the IL-17 receptor only in fibroblasts. And because we see this reduction of chemokines that attract neutrophils, we see significant reductions of neutrophils into the skin. It almost goes to zero. And remarkably, almost all of our transgenic animals had no visible sign of inflammation. So this was a really exciting day in the lab when I first did this because it was just so abundantly clear how important fibroblasts and their recognition of IL-17 is. But this model is a bit artificial, right? We're injecting cytokines into the mouse back. It's not really physiologically relevant. So the next question was, what about Staph aureus? That's what we really care about, right? So we injected our transgenic animals with a MRSA strain of Staph aureus intradermally into the back skin. And we used a reporter strain of Staph aureus. It glows, so we could monitor it non-invasively over time. And qPCR here shows that Staph aureus induced expression of neutrophil chemokines in normal control mice, but this was significantly reduced in our transgenic animals that lack the IL-17 receptor only in fibroblasts. And this led to a staggering reduction of neutrophils recruited to the skin. This was a huge phenotype here. And remember, these cells are really important antimicrobial cells, and now these mice are really deficient in them, right? So they must be at high risk for severe disease, right? Well, that's exactly what we saw. These mice have significantly higher levels of Staph aureus in those lesions, and those lesions are significantly larger. So we really answered the question that we set out to address with this whole thesis, right? Fibroblasts are critical for skin inflammation and host defense, and their recognition of IL-17 is a major determinant of their ability to do so. But as is typical in science, the answer to one question leads to many more, and Rich wouldn't let me graduate so easy, so I had to do some more experiments. So the next question we asked was, are these cells also important in psoriasis? So we're going to shift gears now and start thinking about psoriasis. So psoriasis is not an infectious disease. This is a chronic inflammatory skin disease. It's very common, about 2% of the US population. Here's a picture of plaque psoriasis, the most common form of psoriasis. And you can see this classical presentation of thick, inflamed, scaly skin. And we became very interested in psoriasis because we know that drugs targeting the IL-17 pathway are very effective in treating patients with psoriasis, though they do somewhat increase the risk of infection. So a point that I want to emphasize is that the same IL-17 inflammatory response that protects us against Staph aureus infection can also cause psoriasis if left unchecked. And just like in the Staph aureus context, IL-17 is thought to work by activating keratinocytes to recruit neutrophils. However, in this context, there's no bacteria to kill, and neutrophils still have all of those weapons. So they're kind of bad actors in this context. They're actually damaging us, the host, contributing to cycles of autoinflammation, and the chronicity of this disease. Now, another reason we became very interested in psoriasis was because the role of immune-acting fibroblasts here was also totally unknown. So to test the role of fibroblasts and IL-17 recognition in psoriasis, we used our transgenic animal in a very common psoriasis model, wherein we applied imiquimod topically to the mouse back skin. And here we can see imiquimod induced in normal control mice neutrophil chemokine expression, but this was reduced in our mice that lack the IL-17 receptor, specifically in fibroblasts, shown here both by qPCR as well as immunostaining. And this reduction in neutrophil chemokine expression led to significant reductions in neutrophils recruited to the skin, shown both by flow cytometry as well as immunostaining. Additionally, our mice just looked a lot better. Their disease was almost gone. The skin was less thick, less inflamed, and less scaly than their control mice. So fibroblasts and their recognition of IL-17 is not only important in infectious disease, but is also important in non-infectious contexts, like in this psoriasis model. Now, I've shown you a lot of mouse work today, and mouse models are great for understanding mechanism, how things work, but it doesn't tell us about what we really care about, and that's humans, right? And so the next and final question we asked in this report is, do these cells also play a role in human skin disease? And to begin addressing this, we first analyzed single-cell RNA sequencing data from the skin of psoriasis patients and healthy controls. We performed our network analysis and found that fibroblasts has very strong communication with neutrophils, again here. And using gene set enrichment analysis, we showed that IL-17 signaling was a top upregulated pathway in the fibroblasts during psoriasis. And additionally, fibroblasts were a major source of chemokines in psoriasis. So a very similar picture we're observing here in the mouse as we observe, or in the human, as we observed in the mouse. Now, to understand whether those fibroblast subsets that we identified in the mouse are conserved in human, we looked at the subsets in our psoriasis data, and we again resolved 11 transcriptionally distinct clusters. We found that clusters 1 through 6 on the right of the dotted line highly expressed neutrophil chemokines, whereas clusters 7 through 11 on the left largely did not. And again, we can see CXCL12 very broadly expressed, whereas other chemokines like CXCL1 expressed by a much smaller subset of cells. So again, in the human, different types of fibroblasts that can talk to neutrophils. And to characterize these cells further, we looked at the same panel of known fibroblast subset markers and found that the clusters 1 through 6 highly express reticular and adipocyte lineage markers, but less so papillary and myofibroblast markers. So we've identified an analogous subset of immune-acting fibroblasts that talk to neutrophils in human as we did in the mouse, these CXCL12 positive adipocyte lineage cells. And we confirm this definition again through unbiased methods like GO-term analysis, clusters 1 through 6 expressing genes related to fat cell differentiation and neutrophil migration, but we don't see this in clusters 7 through 11. Additionally, our network analysis showed that clusters 1 through 6 talk to neutrophils much more strongly than clusters 7 through 11. Now, the next question is tricky. How do we demonstrate the significance of immune-acting fibroblasts in humans? In the aforementioned studies, we used a lot of genetically modified mice, right? But we can't do that in humans because it is unethical. So we had to come up with a new approach. And we ultimately settled on recruiting patients that were on and off IL-17 inhibitors, leveraging the fact that these patients are very effectively treated with this drug. And so we first analyzed bulk RNA sequencing data from these patients, and we found that the neutrophil chemokines that are very highly expressed in psoriasis normally were subsequently decreased upon therapeutic blockade of IL-17. This is a bulk RNA sequencing. We don't know which cells exactly are being targeted here. So the next thing we did was recruit more patients and take lesional biopsies from these patients and performed immunostaining. And here we can see neutrophil chemokines labeled in red. And I'm highlighting here in the dermis, the spindle-shaped fibroblast that's highly expressing neutrophil chemokine in untreated patients. But when these patients take IL-17 inhibitors, that expression is lost. And notably, the expression is still retained in the epidermis and keratinocytes following blockade. So somewhat specific here. So these results suggest that IL-17 inhibitors may work in psoriasis by inhibiting immune-acting fibroblast communication with neutrophils. So to summarize this first part of the talk, what we've learned is that in the skin, in the context of Staph aureus infection and psoriasis, the pro-inflammatory cytokines IL-17 and TNF-alpha synergistically activate a subset of dermal fibroblasts that are marked by CXCL12, live in the deep reticular dermis, and are belonging to the adipocyte lineage. And these signals drive the expression of this NF-kappa B cofactor called NF-kBIZ that allows the release of CXCR2 ligands and CXCR4 ligand, CXCL12, to promote neutrophil recruitment into the skin to promote inflammation and host defense. So this work has really fundamentally changed the way we think about how the skin responds to inflammatory triggers. We can now appreciate that keratinocytes and fibroblasts respond to IL-17 to coordinate neutrophil recruitment. And further, our work identifies fibroblasts as a potential therapeutic target for treatment of staph infection and psoriasis. Part 2. In this second part of the talk, I'd like to share with you some exciting unpublished data focused on understanding the role of fibroblasts in Sweet syndrome, which is a human disease of skin neutrophilia. So Sweet syndrome is another chronic inflammatory skin disease characterized by really excessive neutrophil infiltration to the dermis. Here's a classic presentation of Sweets. We have the juicy red nodules on the neck here. And this belongs to a class of conditions called neutrophilic dermatoses, which includes other conditions like pyoderma gangrenosum and pustular psoriasis. So brace yourselves for a little graphic image here, but it's pretty gnarly, pyoderma gangrenosum in particular. So Sweet syndrome is rare, it's yet to be modeled in animals, and therefore it is really poorly understood. We have learned a little bit from case studies, though, and one thing we do know to likely be true is that drugs targeting the IL-17 pathway are probably not going to be an effective treatment option for these patients. So there's probably some other mechanism by which the neutrophils are being recruited and retained in the skin. So we hypothesized that immune-acting fibroblasts, or IAFs, drive the neutrophilia in Sweets, but that they do this through a novel IL-17 independent mechanism. So I've shown a lot of single-cell RNA sequencing data today, and while this is a very powerful technology, there are a few limitations, and one is that it requires fresh tissue. And this is really tough to get when you're working with a rare disease. However, a new single nuclei technology developed by 10x Genomics works on formalin-fixed, paraffin-embedded tissue. And this is really important because it allows us to investigate archived clinical samples. Whenever a patient comes into the clinic for diagnosis, they get a biopsy taken and it gets put into FFPE, and that can be stored indefinitely. So we have biopsies stored in FFPE, and we used this technology on a cohort of patients with neutrophilic dermatoses and healthy controls. And with this approach, we recovered all the major cell types in the skin and a nice about 1,000 genes per cell and 1,600 transcripts per cell. So not too bad for working with old fixed samples. And we can start using these data to make new discoveries. And here, looking at composition, we can see, we knew that neutrophil myeloid cells were increased in neutrophilic dermatoses, but now we're learning that lymphoid cells are also increased, as well as plasmacytoid dendritic cells and dendritic cells, for example. Now, to understand which cell types may be driving the inflammatory response in Sweets, we looked at the differentially expressed genes between every single cell type in Sweets compared to every single cell type in healthy skin. And we found that the fibroblast was the most activated cell type in Sweet syndrome. So we looked at the fibroblast subsets in our data and resolved 7 transcriptionally distinct clusters. And the one I'd like to highlight today is this FB6. And this is marked by these IFI genes. And IFI stands for interferon induced. And interferon is another pro-inflammatory cytokine, but this one's mostly associated with antiviral responses and some autoimmune conditions like lupus, but not neutrophilic dermatoses. However, we found that this FB6 was entirely specific to Sweet syndrome. We didn't find it anywhere else, just in sweets. So we've identified a new biomarker of sweets, these interferon-activated fibroblasts. Now, we next validated our single nuclei findings by performing a large independent cohort study, about 50 different patients with neutrophilic dermatoses per group here, and performed bulk RNA sequencing on also FFPE tissue blocks. And we first deconvolved these data to validate our single nuclei findings. And we again see not just increased neutrophil myeloid cells, but also lymphoid cells, PDCs, and DCs in the neutrophilic dermatoses, so validating our single nuclei findings. We next performed differential expression analysis between the neutrophilic dermatoses and the healthy control skin, and then identified the 651 genes that were uniquely regulated in Sweet syndrome. And we performed pathway analysis with these genes and found that one of the top pathways unique to Sweet syndrome was HPV infection. And remember that interferon goes hand in hand with viral infection. So many of these genes that are specific to Sweet syndrome are interferon-induced genes. So we see these genes highly expressed in Sweet syndrome, not just relative to healthy controls, but also pyoderma gangrenosum. So using this independent large study, we validated our single nuclei findings. Now, the next question is, where do these interferon-activated fibroblasts that we've identified in Sweet syndrome reside in the tissue? Where do they live? So another limitation of single-cell RNA sequencing is that it does not provide any spatial information. We know what all the cell types are and all the genes they're expressing. We don't know where they are. However, another new technology, this one called CosMx, developed by NanoString, provides spatial context at very high resolution and very high plexity, 1,000 different genes we can measure at once. And this also works on FFP and will allow us to study our archive samples. So we use this approach on a cohort of patients with neutrophilic dermatoses and healthy controls. And with this approach, we do the same sort of pre-processing as in the single nuclei, but now we can take those clusters that we get and project them back onto the skin to see where they are in space. And we can start making new discoveries. For example, we can see that the basal or lower level of the epidermis in gold is transcriptionally very similar between the 3 conditions. However, the suprabasal or upper epidermis is transcriptionally very different between the 3 conditions. With these data, we can also perform more sophisticated analysis, like niche analysis. And here we can divide the tissue up into different regions or niches, and then we can compare the fibroblasts that are in one region to the fibroblasts in another. And here we can show by doing differential expression analysis between the fibroblasts in the upper dermal niche in green, niche 1, to the lower dermal niche 2 in purple, that an interferon-induced gene is highly expressed in the lower dermal niche, IFIT-M3. Therefore, we've used this technology to locate those interferon-activated fibroblasts in sweets to the deep reticular dermis. So finally, the last question here is, what function is served by these interferon-activated fibroblasts in Sweet syndrome? What are they doing? And to address this, we performed an in vitro functional assay. And here we took primary human dermal fibroblasts from 50 different donors. This is a very big study. And we stimulated them with interferon gamma, interferon A2, and TNF alpha, and then performed bulk RNA sequencing. And as expected, the interferons induced interferon-induced genes, though we did not see this with TNF alpha as expected. However, we were very surprised to find that interferon A2 induced expression of neutrophil chemokines, and it did so to the same extent as TNF alpha. So these results suggest that immune-acting fibroblasts' recognition of type 1 interferon like interferon A2 may drive these cells to recruit and retain neutrophils and promote pathogenesis in Sweet syndrome. So to summarize the second portion of the talk, we're really using these cutting-edge transcriptomic technologies on archived fixed samples to provide unprecedented insight into these rare, poorly understood diseases. And using these approaches, we've identified Sweet syndrome, a prominent interferon signature in this disease, and we've particularly located it to the fibroblasts in the reticular dermis. And functionally, we've shown that interferon, type 1 interferon, like interferon A2, activates human dermal fibroblasts to express neutrophil chemokines. So these data suggest that interferon-activated fibroblasts may contribute to sweet syndrome pathogenesis and may represent a therapeutic target for treatment. In conclusion, I hope I have taught you today that fibroblasts are not merely structural cells. These cells are incredibly diverse, and some of them are immune-acting fibroblasts, or IAFs. In the first part of the talk, we learned that immune-acting fibroblasts in the skin protect us against Staph aureus infections, but also contribute to psoriasis pathogenesis. And very important to this is their recognition of the cytokine IL-17. In the second portion of the talk, I share with you purely human data showing that immune-acting fibroblast recognition of type 1 interferon may contribute to the pathogenesis of sweet syndrome. And while my work is really focused on demonstrating the significance of fibroblast recognition of IL-17 in the skin, we propose that fibroblasts are key integrators of many different signals, both host and microbial derived, and that in turn that these cells orchestrate many aspects of immunity, not just neutrophil recruitment. And further, we propose that there are different types of immune-acting fibroblasts that are doing these functions, and that these cells exist in other tissues like the gut and the lung. So I'd like to emphasize that before my thesis, we did not appreciate the role of fibroblasts in skin infection and inflammation. However, now after my thesis, we can confidently say that fibroblasts are absolutely critical for these processes. However, there are still many outstanding questions. For example, what are the role of these cells in other inflammatory contexts in other tissues? What about type 2 inflammation in the skin and atopic dermatitis or neutrophilic asthma in the lung? There's a lot of questions about how these cells are integrating all of these signals, and more specifically, how is NFKBIZ induced in these cells, and how does it promote synergy? And then we're also very interested in understanding this new subset of cells that we've identified and really defining it better. What is its development, its fate, and is it plastic? So we know that these cells express adipocyte lineage markers, but are they destined to become fat cells? And then in terms of plasticity, can a CXCL12 positive cell become a negative cell, or vice versa? Still open questions. And then finally, a more long-term goal is to understand how to therapeutically target these cells specifically to develop treatments with better efficacy and fewer side effects. So with that, I'd like to thank the people who helped me with this work. The first part of the talk was done solely in the Gallo lab, so huge thank you to my mentor, Rich, for his guidance. A big thank you to Carlos for doing literally all of the mouse genotyping and breeding. Saved me so much time. And also need to thank Fong for doing the bulk of the mouse and human tissue immunostaining. And the second project was done in collaboration with the Johnson lab at the University of Michigan. So I need to thank Johan for being a very generous collaborator with resources and data. And then need to thank the Derm Clinic for providing all of the human samples. And then also just want to thank the entire Gallo lab, who was maybe not on this project, but who asks great questions during lab meeting and listens to all my talks and provides feedback. Many of them are here today, so thank you all. And then I need to thank my funding sources too. I was very fortunate to have funding for the entirety of my PhD. So the first portion of my PhD, I was funded by it's a long one here, the UCSD Gastroenterology NIH T32 training grant. And then the latter portion, I was funded by the NSF GRFP. And a big thank you to ARCS for their additional financial support. And thank you all for listening. I'm happy to take any questions and discuss. Nikki. Do neutrophils talk back to the fibroblasts is the question. So I have not done that experiment, but I'm willing to wager that they do, because neutrophils highly express some cytokines like IL-1 beta. And in some of our in vitro experiments, we stimulate the fibroblasts with IL-1 beta, and they make a lot more neutrophil chemokines. So I think that, yes, there's this positive feedback loop there. Those are great questions and stuff we are actively working on. I think we're really just at the tip of the iceberg here in understanding what these cells are doing and their significance. But yes, those are great questions. Clinton. So those are great questions too. So my project, just like I said, tip of the iceberg, I think, understanding how this new subset of cells changes with age and with sex and so on, with like obesity, all these factors are something we're going to work on. But what I can tell you is the work that the postdoc did that first inspired a lot of this work, Ling Zhang, with the reactive adipogenesis thing, she's worked a lot on understanding the impact of age. And we know that when you get older, you're at more risk for infection. And what she's shown is that reactive adipogenesis is suppressed in the elderly in a TGF-beta dependent manner. So I hypothesize that their ability to recruit neutrophils is probably also impaired with age, but we haven't done those experiments yet. But great questions. So that's very long term, above my pay grade for sure. In terms of therapeutics, what do I imagine? So in terms of like, we have some drugs right now that are really effective, right? Like these IL-17 blockers in human psoriasis patients. But there are some caveats of these. They do increase the risk of infection a bit, and so I think designing more specific drugs is going to help us, if we can target the cells specifically and, not just target cells all over the body, but maybe just the skin fibroblasts. So this is definitely more of a biomedical engineering question, but in terms of targeting cells specifically, there's bispecific antibodies that are a popular thing these days, where you can not only target the cytokine, but also another molecule. So we could have an antibody that targets fibroblasts attached to an antibody that targets IL-17 and touch just the fibroblasts specifically. That would be like a systemic treatment probably, but ideally we would be able to put something just on the skin, right? And not have it go all over the body. So like, I think some sort of absorbable compound would be the best approach. Just in the chat from Taylor. Okay. In one of your seq data sets, I think staff induced TSLP and IL-33. Do you think these fibroblasts are an important source of these type 2 alarmins, protein level? Why, I sure do, Taylor. These are things that I've been trying to convince Rich to buy the conditional knockout mice for me to play with. Yeah, and I'm not sure exactly what they're doing in the context of Staph aureus. But I do believe, yes, that they are significant sources of these molecules as well, which is, Taylor is an allergist and immunologist, and I was in his lab previously. I'll talk about this, but we in the lung think of epithelial cells as being the major source of these alarmins like TSLP and IL-33. But I think, even in the lung, that fibroblasts may be a contributor there as well. And that's something that I'm interested in working on with you, Taylor. So we'll talk more. Right. Yes, this was Cosmics, it was called, spatial transcriptomics. Yeah. So are you asking if we can detect IL-17 with this technology too? The effects of. So it's tough, right? Because we're working with a single sample in one point of time, right? But if we were to do maybe like a time course study, like after their therapeutic blockade, get time series data, we can better understand, the effect of IL-17 on the architecture of the skin and the fibroblasts. Is that toward the what you're getting? Yeah, I think it's tough with humans, right? Because in mice we could do these mechanistic studies, but in the human we're limited to correlational work.